Hi, this is Jens Soering, and this is the second part of a 5-part series on DNA in the Haysom murder case. The subject of the second part is whether the samples at issue might have been mixed or contaminated. That's an important question because if they were mixed or contaminated, the results might not be reliable. This issue was first raised 7 years ago in the fall of 2016. On September 11th, 2016, the Richmond Times-Dispatch published an article about this, which I've linked up below. I always link up all of these things so you can check for yourselves whether I'm telling the truth. On September 11th, 2016, the Richmond Times-Dispatch interviewed Betty Lane Desportes. This is an attorney who specializes on forensic evidence and specifically DNA. So it's somebody worth listening to. And in this article, she first raised the question, are these test results reliable or could there be mixture or contamination? In order to follow up on this, my wonderful attorney, Stephen Rosenfield, found 2 DNA scientists who were willing to look at all of this basically for free. Their names are Professor Moses Schanfield from George Washington University and Professor Tom McClintock from Liberty University. These are 2 of the most respected DNA scientists in the whole United States. I've linked up their CVs below so you can check. Each of them has testified in over 100 trials as a recognized expert witness on DNA. They're just the top of the field. And they looked specifically at the question of whether there was mixing or contamination in these samples. And I've linked up their reports below. There are 2 from Mr. Schanfield and 2 from Mr. McClintock. And they specifically excluded that there was any mixture or contamination. They said that the samples that are at issue here come from a single contributor. Don't believe me. Read the reports yourselves. And I've also included articles where they were interviewed by newspapers in which they confirmed this. And I've linked up a press conference where Professor Schanfield testified at length about the DNA tests. Actually, what they specifically found was that there was no evidence of mixture or contamination. Therefore, you cannot just assume that there must have been. You have to go with the evidence you have. And if there's no evidence of mixture of contamination, then you must assume that there wasn't any. One of the things that they found really significant is that back in 1985, way back then, when they did the blood group serology tests, they were able to find mixture contamination in one sample. It's called 13K. It's not one of the 4 samples that's important, right? This is a completely different sample. This just proves that back then, they were able to detect mixtures. Here I'm showing you a picture from the 1985 serology report, from the tables at the back. You can see for 13K there are 3 numbers there instead of 2. That means it's a mixture. The scientist who performed these tests, Jane Burton, testified about this at my trial. Here's a picture from the trial transcript. She says, yes, that means that 13K is a mixture. And it's the only sample like this where she was able to detect a mixture. All of the important samples did not have a mixture. And now, please take a look at the DNA report. I'm showing you the table here at the back. And you see that for all those different test results, there are always 2 numbers in each of the columns, not 4. And that suggests no mixture or contamination. Let me explain that briefly. We have to go back to biology class. What they're testing for in DNA tests are chromosomes. And chromosomes, each chromosome, that's each one of those columns, each chromosome has 2 alleles. That means 1 from your father and 1 from your mother combine to make a chromosome. That's why you see 2 numbers. 1 from mom, 1 from dad, for every one of them. Where there's nothing, they didn't get a result. These are partial profiles. But where you did get results, you see 2. If there were a mixture of 2 different sources in this sample, you would have 4 alleles, naturally. Because for each one of the 2 sources, you would have a mother and a father, and then for the other one, you'd have a mother and a father, and you would have 4 alleles. But in none of these columns do you see 4 alleles. You only see 2. And that's explained in Professor Schanfield's and Professor McClintock's reports. But that's how you can tell. There is no evidence of a mixture. That should have really been it. But then I was released in 2019, and after my release, a British police officer who was involved in the case said that there must have been a mixture or contamination. Because if there weren't, then I'd be innocent and he'd look really, really bad. But again, this is a British police officer who may be a good police officer, but is not a DNA scientist. And everybody's entitled to opinion, but he is simply not an expert, and he would not be allowed to testify in court as an expert witness on DNA. Only Professor Schanfield and Professor McClintock would be allowed to testify as experts on DNA, because DNA is one of the most sophisticated and complicated sciences in existence. And this British cop might be a nice guy, might be a good cop, but he hasn't been trained in that. Think about it. If you need brain surgery, are you going to go to a car mechanic or are you going to go to a brain surgeon? You're going to go to a brain surgeon. Same thing here, right? This is DNA. You don't go to a cop to find out what the actual facts are. You go to 2 sophisticated, fully trained professors of genetics from some of the best colleges in the United States, Professor Schanfield and Professor McClintock. But because this criticism was put out there by this British police officer, whose reputation is riding on all of this, by the way, I decided to ask Professor Schanfield and Professor McClintock to go back to the lab and look at the raw data. And it turned out, in the meantime, Professor Schanfield had died during the COVID epidemic, sadly. But Professor McClintock was still alive and he was willing to do this. And he actually went to the Virginia Department of Forensic Sciences, and they actually went way out of their way to help him. They actually provided a staff member to assist him to look at the raw data from the DNA tests performed in 2009. Nobody had looked at this between 2009 and when Professor Schanfield went to the lab in 2022. He's the first person to do this. And he looked at the raw data and he was able to confirm once again that all of the samples at issue came from a single source, single contributor, no mixture, no contamination, from the raw data that nobody else has looked at. I've linked up his report, his follow-up report, below so you can check this out. This is from last summer, August 2022. So at this stage, there's really nothing more to say about the theory of mixture or contamination. You cannot exclude it completely, but there is no evidence that it actually happened. And that's not me saying it. It's 2 of the top genetic scientists, and one of them even went through the trouble to look at the raw data from the DNA tests. And I think if you're going to be fair about this, you have to conclude there is no mixture, there is no contamination. You can trust these test results as I explained them in part 1 of this series.